immediately find dramatic claims about extending lifespan and preventing chronic disease. Yet the most staggering of those claims share a surprising common denominator. They have never been proven in humans. The public discourse around fasting treats the science as one massive, unified monolith. In reality, the research exists as two completely separate bodies of evidence. The first body of evidence consists of controlled clinical trials conducted on humans, which focus strictly on measurable metabolic health. The second body of evidence relies exclusively on animal models, testing extreme longevity and cellular repair. When pop science headlines promise that skipping breakfast will trigger total cellular renewal, they are simply lifting the spectacular results of those animal studies and marketing them as guaranteed therapies for human patients. To find the actual clinical utility of fasting, we have to pull these two categories apart. We need to build a framework that separates human circadian biology from animal cellular repair. Learning to isolate clinical reality from preclinical hype protects you from adopting extreme restrictive dietary habits based on entirely unproven premises. We start with the human side of the evidence. At the Salk Institute, circadian biologist Sachin Panda and his research team have spent years studying not just what people eat, but when they eat it. This 24-hour circular clock maps our evolutionary baseline. Human metabolism evolved to process food within a roughly 12-hour daylight window. But when Panda's team used smartphone apps to track ordinary eating patterns, the data surprised even the researchers. Participants were routinely snacking late into the night, pushing their daily eating window out to 15 or 16 hours. This expansion collides directly with circadian biology. Organs like the liver and pancreas do not wait passively for food to arrive. They operate on a rigid internal daily timer. Metabolic processes like insulin sensitivity and fat processing peak in efficiency during daylight and plunge at night, independent of calories consumed. Expanding an eating window to 16 hours forces organs to process nutrients during their lowest efficiency periods. To test this, researchers introduced a clinical intervention called time-restricted eating. The protocol simply compresses the daily eating window back down to a daylight-aligned 8 to 10 hours. Crucially, these trials did not ask participants to change what food they ate, nor did they restrict how much they ate. The only variable altered was the timing. Modern eating schedules frequently force organs to process nutrients outside of their peak efficiency window, creating a persistent mismatch between behavioral habits and cellular timing. When human subjects adhered to time-restricted eating, the clinical trials returned measurable, verifiable outcomes. The results showed better blood sugar control and minor weight loss. These benefits are real, but they are distinctly modest. Furthermore, these improvements appeared most prominently in individuals who began the trials with existing metabolic risk factors, like prediabetes. The data paints a different picture than the grand claims found online. This research validates a specific daylight eating habit rather than a total metabolic overhaul. It is also worth noting that these controlled human trials are still relatively short in duration, and research teams are currently working to replicate them at a larger scale. The highest quality human evidence establishes fasting as a tool for modest metabolic regulation, providing a realistic baseline for what a patient can actually achieve. In 2019, physician researchers Rafael DiCabo and Mark Mattson published a widely cited review in the New England Journal of Medicine, pulling together the totality of existing fasting data. This graph illustrates the metabolic switch. As fasting hours accumulate, readily available blood glucose is depleted. Once glucose drops, the body pivots, producing ketones for energy. Alongside this ketone production, extended fasting windows cross a threshold, triggering deep cellular repair processes. Clinicians highly valued DiCabo and Mattson's review precisely because it approaches this threshold with extreme caution. The authors explicitly state that the dramatic disease prevention and longevity claims featured in their own paper were sourced almost entirely from rodent studies, not human trials. Human fasting trials cannot currently validate claims of disease prevention because they are simply too short to track chronic illness outcomes over a lifespan. DiCabo and Mattson specifically called for decades of rigorous, longer-duration human research before any doctor should treat fasting as a medical cure-all. The metabolic shift to ketones is a proven human mechanism. But taking that acute metabolic shift and projecting it forward as a guarantee of long-term disease prevention requires a leap across decades of missing data. That brings us to the second pillar of evidence, the staggering claims of extreme cellular repair and massive lifespan extension that dominate internet algorithms. This data often stems from labs studying aging at the cellular level, including high-profile caloric restriction research by scientists like David Sinclair. The data produced by these labs is striking and properly replicated, but it is strictly confined to lower organisms like yeast, worms, and fruit flies. Much of this research focuses on specific compounds that activate sirtuins, proteins theorized to play a crucial role in regulating cellular aging. Yet, this cellular mechanism carries a critical constraint. It is occurring inside a different species entirely. Lab mice live in sterile environments with meticulously controlled genetics. The biological reality for humans is far more variable, occurring within chaotic, uncontrolled environments. This horizontal scale highlights the biological translation problem. Researchers are attempting to map interventions from a 2 to 3 year mouse lifespan onto an 80-year human lifespan. Running a proper aging study on humans to confirm these effects would require locking thousands of people in controlled environments for multiple decades. Without that control, translation consistently fails. Multiple promising compounds that drastically extended mouse lifespans have shown much weaker effects or failed entirely in the limited human trials we do have. The mouse data is real, but applying it to humans misrepresents the reality of how cellular biology scales across species. To accurately evaluate any future claim you see about fasting, we can map the current medical evidence onto this split-screen matrix. In the left column, human clinical trials confirm that time-restricted eating provides modest, refining metabolic benefits for some people. In the right column, animal models strongly support the occurrence of deep cellular repair and massive longevity extension. While the mechanisms on the right are biologically plausible for humans, the evidence has not crossed the boundary line into human trials. Acknowledging this gap does not mean fasting is debunked or useless. It means fasting research exists at wildly different stages of maturity, depending entirely on which specific claim you are investigating. You can apply a simple clinical heuristic. Whenever you encounter a dramatic headline about fasting, stop and check the citation species. Until long-term human clinical trials mature, evaluating the biological distance between yourself and a lab mouse is the only way to read the science accurately. Stratifying evidence in this way replaces a reliance on headlines with a rigorous, investigative approach to clinical truth.
And nothing here should replace a conversation with your own doctor, especially if you have a metabolic condition, take medication timed around meals, or have any history of disordered eating. What the research actually supports, cleanly, is a habit of reading the specific claim, not just the word fasting checking whether a given benefit comes from a controlled human trial or a rodent longevity study. Because right now, those two categories of evidence do not yet say the same thing. Follow along for more research broken down the way the actual papers present it, not the way the headlines built on top of them do.